Hi, everyone. For those who missed it, my name is Megan Lee, and I'll be your moderator today throughout this webinar. We will have two speakers today, Dr. Anish Acharya, the CCO of Molecular Instruments, and Dr. William Damsky, an assistant professor of dermatology and pathology at Yale School of Medicine and the innovator behind the groundbreaking LDTs that utilize HCR Pro to characterize inflammatory skin diseases. After the presentations, we'll move into the Q&A portion where Dr. Jason Ramos, the Senior VP of Product Excellence at BioCare Medical, will join Dr. Damsky and Dr. Acharya to help answer your questions. Thank you for being here, and we'll get started by inviting Anish on stage.
Okay, I think that might be working now. All right, thanks a lot, Megan. Really excited for this webinar. This is a topic that for a lot of us at MI who were part of the original crew sort of developing this at the bench at Caltech not that long ago to come to where we are now, where thanks to the work we'll see from Bill and Jason's team as well, that some of these assays can be deployed for clinical use. It's humbling and exciting, and I'm really glad we can do this. And obviously representing MI, we're proud of our contribution here, but really we recognize that we're equal legs of a stool, along with of course Bill and then Jason's team at BioCare to sort of build the hardware that makes a lot of this possible. So in terms of the plan for today, I think I'm only on stage for 10-15 minutes. I'm going to go through just some basics about how HCR works and what we generally offer, and then we'll lead into Bill's part. And probably the best thing I can say about Bill is that when he started working on this, we actually didn't even know. So it was kind of an off-the-shelf product that he was able to validate and implement in the way that he'll describe later on. So we're Molecular Instruments. The core technology that powers all of the things we do is HCR. And so I want to start by showing a little bit how HCR works. I like to share with people that HCR is not an imaging method. It's not about ISH. It's just a general purpose DNA-based amplification tool that our lab invented when we were still at Caltech, and we then applied to RNA-ISH for imaging. So here's HCR. It comprises these three components. Everything here on this slide is DNA. So H1 and H2 are what we call DNA hairpins, and we engineer these sequences so that they don't directly interact with each other. So if you mix H1 and H2 on a tissue or in liquid biopsy or whatever the sample may be, they remain closed as H1 and H2 monomers. And the sequence that mediates their interaction or triggers their interaction is called the initiator, or here it's labeled I1. I1 is not a target-specific sequence, but rather a sequence we designed to work with this system of hairpins. So what an I1 can do in situ is bind to this free toehold of H1, and through a process called branch migration, open up the hairpin, so you form this kind of I1-H1 complex. And now, on the H1 part of this complex, you have this freed single-stranded region that previously was double-stranded and trapped and couldn't interact. And now this freed part can do something similar to an H2 molecule, where it binds to the single-stranded toehold and through branch migration opens up the hairpin to form this kind of complex. And then the cool trick we do is that this freed sequence of H2 is actually identical to the initiator. And so in situ, you get this back and forth addition of these monomer hairpins, and you form this long polymer in situ. So for users, this is great because I can show this slide, and if nobody understands how any of this works, it's okay because it's a very simple one-step reaction. Actually, for the automated kits, which we started shipping a few years ago, you don't even have to mix the hairpins. It just comes ready to use in a container. This is how we implement background suppression as well. And since we started, HCR is the only ISH method out there that has background suppression both in the probe binding stage or the target detection stage, as well as the amplification stage. And the reason we call this background suppressing, I should say, is because most of the sequence of the hairpins is double-stranded and can't trigger unless there's a growing polymer. And then some cool things which we enable a lot of the features that we're getting known for more and more, like being protease free and sort of pioneering that workflow, is that this whole reaction is very, very mild. So it's isothermal at room temperature, doesn't use an enzyme, and is optimized to work in just salt buffer. So really, really mild on precious samples. And over the past 10 to 15 years since we first started, we've seen HCR amplification catalyze lots and lots and lots of progress for RNA-ish. I'm going to show a little bit about what some of our users can do because we actually have this large foothold in the developmental biology community because we always try to cater to sample types that are not necessarily popular or model organisms just yet. So this is the second component of the kit that forms a product for RNA-ish. So we went through the amplification or how you generate signal, and this is the probe. So we call these hi-fi probes because they work really well, but also because they're based on this fractional initiator design, which uses a pair of probes to bind to a specific binding site in order to form a full initiator sequence. And this is how we implement background suppression in the probe binding stage. So if you happen to design a binding site that is not optimal and one of the probes in a pair sticks non-specifically in your sample, it doesn't carry enough of the initiator to trigger amplification. And in situ, you get really, really good performance. And especially as we've launched this biopharma and translational and clinical focused product, which I'll show a little bit more later, we've seen that these probes really lead the industry in sensitivity, specificity, and something that's really critical for us is auditability, which may or may not be a real word, but for us means how easy is it to check? Is your ISH signal correct? Or is it something that shouldn't be binding there at all? And last year we talked a lot about how do we achieve clinical grade performance, and that's not going to be the topic of today's talk because we'll have Bill come on and actually show clinical grade performance. But on our end, we really try and make the kits as easily validated as possible. Okay, so I want to show one little analogy because if you know me, I love my analogies. And one of the most, I suppose, popular reasons we're known is we have this assay that started its life as being protease free and still is. And we've seen, especially over the past couple of years, as we've gained so much traction, some of our competitors also try and claim protease free, but I like to always show why we're protease free. So this is a street in San Francisco. If you don't recognize it, it's called Lombard Street. Next time you're in San Francisco, I recommend a visit. I don't know if this is true or not, but it's called the crookedest street. I don't know what jurisdiction that is, but it's very, very windy. You can see this red car here in the left for some scale. It's really steep, but the aerial view kind of eliminates that effect. But we can kind of think about tissue this way, that you have a really, really windy street. You have these windy channels that go through your tissue, and whether you're doing ISH or IHC or whatever your stain of interest is, the whole name of the game is, can you diffuse reagents through your tissue easily, let them get to where they need to be, find their targets, find their reactive element, and then wash out in a comfortable way. And so for us, when we were engineering HCR, a lot of the constraints we had were around how do we work with really thick sample types like whole mount embryos. And then the second one, as we think about being clinical grade, it's if we want ISH to be at the level of IHC, we need to make it so that the sample is identical to what people do for IHC. So the goal was always don't do any additional sample prep for ISH that you wouldn't do for IHC. So that obviously precluded protease, but also precluded any kind of other digestive element. So today the only thing that we require for thin tissue, FFP tissue, is standard retrieval. And so I want to show a little bit about why. I've drawn all of these boxes to appropriate scale. But if we anchor the size of the typical kind of reagent that a user is trying to flow through a tissue at an antibody. So a typical antibody is about 150 kilodaltons in mass. And if we say that's like a Honda Fit or a typical compact car, it can pretty easily navigate down this street, although you may not choose you want to do it. Now, with HCR, the single biggest molecule we're trying to flow through a tissue are those hairpins, H1 and H2 that I showed before. They're the same size, and they're about 35 kilodaltons. And so actually a fraction of the size of anything you need to flow through for IHC. And this is really the magic of HCR. It's not some black box. It's a very simple thing, which is because we're building these big polymers from very, very small background suppressed elements, we can work with all kinds of milder sample prep compared to some of the legacy-ish methods that are out there. And in terms of those, the most popular ones are based off of an old technology called branch DNA amplification that focuses on building these big trees with big, bulky building blocks. And when you look at some of those molecules, because you can't build a big tree without a big tree trunk, they can be upwards of 300, 400 kilodaltons of mass. And to tie back to the analogy, there's no way this bus is getting down Lombard Street unless you do some pretty significant digestion of the street. Whether that's called protease or whether that's called something else, it's really not the point. The point is the underlying technology. So today we have two product lines. One we call HCR Gold. This is our expression for what a modern, simple, elegant assay for multiplexed fish looks like. So today it supports up to 10-plex parallel imaging. Truly any sample. We don't mean any mouse or human section or any tissue section, truly anything. And I'll show a couple of slides about what that means. And increasingly, we're working with more and more of the traditional spatial biology companies to establish these partnerships to bring, really best-in-class RNA imaging to those typically protein imaging assays as well. So here's an example of the kind of image that a user can get with the HCR Gold era of products. And so this is a whole mouse embryo. And so not a tissue section. This is a 3D sample that is millimeters thick at its minimum dimension, using the same two-step protocol that we already went through. I always like to show one of our more exotic submissions from our users, mostly because this really highlights how portable the technology is. I think we can say with credibility that there's no way for us to develop a protocol for sea urchin larvae, but our users are really able to get these reagents kind of working in all these non-model or emerging model organisms. This is a poster that we presented at AACR just recently with Brooker Spatial Biology. And it was a project at St. Jude Children's Hospital trying to do sort of multiplex detection of protein and RNA, tracking a CAR T, like in real patient samples from children with brain cancer. And so we're really excited about, of course, the project, but then also building a technology platform that can be used for all kinds of really compelling applications. And to date, we have over 5,500 direct citations. So this is actually not publications that use the technology. That number is significantly larger. These are direct citations of the underlying HCR technology platform. So we're really excited about all the different things people have been able to build using these base reagents. So the second product line we have is called HCR Pro. This is what began its life as our automated product that was designed for more translational and clinical use. And today is serving actually all kinds of users along the sort of the discovery spectrum. So today this supports up to 2-plex, and it's we call it a flexible 2-plex because one of the things we had to develop to support bright field imaging was the ability to deposit chromogens. And so this ends with an AP or an HRP for a user, and a user can do 2-plex with AP HRP or 2 HRPs and do chromogenic or fluorescent imaging. And it's always protease free. So there's not multiple options on how to do this. Because we know users are always working with thin tissue, we can actually just make a unified protocol that does not require any digestion beyond typical IHC sample prep. Okay, I want to show a couple of images. Here's one showing our 1-plex bright field image on an Encore Pro, which is what Bill also used as the hardware for his assays that he'll talk about in a few minutes. This is with mouse duodenum tissue and just really gets really clean, good performance. Here's an example of 2-plex, but using fluorescence. So using our HRP kits, you can just swap out the chromogen for a depositable fluorophore, TSA-driven fluor, and also gets really, really good performance. And while on the academic side, we like to see publications because it's validation that the products are worth users' time. On the biopharma side, we tend to get fewer publications, but more quotes like this that really, really matter. So, we've never been a company about just promoting pretty pictures and multi-omics and all of this stuff, but rather, how do we drive people to be able to get work done better? So this is one example of a senior scientist in biopharma who just found that because the kits validate quicker, they can get started faster with their actual work. Another different user at a different biopharma company told a story that I think we hear a lot, which is he has an internal pathologist who's uneasy when they're looking at new technologies, and we empathize. But when they see side-by-side versus legacy methods, people are just generally very impressed by the clarity of staining, the integrity of tissue, because we don't use any harmful digestive enzymes to make this work. So today we have over 6,200 users around the world and over 400 biopharma sites. So we're really excited about how quickly the Gold and Pro products are being used, and we're getting good feedback about how well they're working. And then the final thing I want to talk to just to segue into Bill's section is, last year I mentioned we talked about this a lot. We had workshops at NSH, we talked about it at HCR and SITC, and we'll continue to. But really our North Star is how do we attain clinical grade performance for not just our products, but for ISH more generally as an industry. And we have a sort of simple 3-spoke framework for how do we think about that. And the first is performance. We always have strived to have the best performing assays. I think we can credibly claim based on real user feedback that we have best in class performance with the Pro kits and the Gold kits. Second is turnaround time. So since day one, we've always tried to make these kits faster to run than anything else out there. And really the goal is not to be the fastest ISH method. The goal is to be on par with IHC, which can sometimes be as quick as 60, 90 minutes. So it can be done sort of at point of care. And then finally is cost. We live in the same world too, but our approach is technology is meant to get cheaper over time. It's not meant to come out year one and then get 5% more expensive as it supposedly tracks inflation. And so this is an area where we're proud to have the lowest per sample cost, and is an area where we're also investing how do we make this even lower to drive clinical adoption. And the final thing I just want to add before we segue is sort of the other spoke of this stool that I mentioned before, the other leg of the stool, which is our partnership with BioCare. This is actually an announcement from over a year ago. We collaborated with BioCare to sort of enable all their assays to run in a pretty hands-free way on the Encore Pro, and that's really what led us to a Bill as well, who's running everything you'll see with ISH coming next will be on the Encore Pro. So I think that wraps up my section. I'm gonna transition to Bill, and then we'll answer any questions, I suppose, towards the end.
All right, Anish and Megan, thank you so much for having me here today. Hopefully you can hear me okay. So I'm a dermatologist at Yale University. I see patients with inflammatory skin diseases. I also am a practicing dermatopathologist, and it's sort of that intersection of my clinical involvement that has led me to develop these tests, which I'll tell you more about today. So I'm going to tell you about work that we've done in 2 diseases called psoriasis and eczema or atopic dermatitis. Like many fields, dermatology is a field that's really exploding in terms of the translational revolution. So you can see listed here are very common inflammatory skin disease like psoriasis and atopic dermatitis, which we'll focus on today, but other inflammatory skin diseases which also have molecularly targeted therapies. And really what you can see, especially for psoriasis, is that there's so many different medications that are FDA approved with so many different mechanisms of actions. Uh, how does really a clinician choose which therapy is best for their patient? And that will sort of be the crux of the talk today and sort of this idea of personalized medicine. But I also really like to make the point that this is really the tip of an iceberg. There are so many more inflammatory skin diseases in dermatology, and undoubtedly there will be additional medications which are improved, and often, undoubtedly, medications with differing mechanisms of action for the same inflammatory disease. And so, really, how do we prioritize the best medication for an individual patient? So this is a way that I like to start this talk, and it's really a very fundamental question. So let's take atopic dermatitis as an example, and I'll refer to atopic dermatitis and eczema sort of interchangeably throughout the today's talk. But if we have a medication that treats this disease, and the medication works in a certain way, why don't all patients respond equally? Why do some patients respond to the medicine and other patients don't? And I think this is really well illustrated by this phase 3 study of a large number of patients with eczema or atopic dermatitis. Really, IgA reduction is just improvement in their skin disease. But what we look at here is two things. One, only about 36.5% of patients actually reach the primary outcome. So why isn't it 100% if these patients all have the same disease? And when we look at this study, which compared dupilumab, which blocks IL-4 receptor alpha, IL-4 and IL-13 signal via IL-4 receptor alpha, why don't the patients all respond, and why is this variable response? In contrast, JAK inhibitors, which JAKs are proteins that block a number of different cytokines, IL-4, IL-13, and others. Although the trends aren't always statistically significant when we're looking at dupilumab in green here at 16 weeks after initiation of drug. But when we look at the JAK inhibitor, there seems to be a non-statistically significant increase in response rates. And so why is that? Are other cytokines involved? And when you read the textbooks, IL-4 and IL-13 really are the canonical cytokines in atopic dermatitis or eczema. So our group really got interested in this question. There's obviously a number of different possible explanations in terms of why patients don't respond the same to the same medication, even though they have the same diagnosis. One possibility is dosing. Maybe some patients require a higher dose. Maybe the outcome isn't right. So I showed you one outcome in the clinical trials. There's no one perfect way to measure how much a patient improves or doesn't. Maybe there's pharmacokinetic or pharmacodynamic considerations, maybe genetic variation matters. But really what I want to focus on today is this concept of disease endotypes. What are disease endotypes? Well, disease endotypes, the way that I define them, are a subtype of disease defined by a distinct pathophysiological mechanism at the molecular and or cellular level. They aim to explain why disease occurs in a particular way in different patients. And I'd like to extend that to say why patients may or may not respond to the same targeted therapy. So when we're thinking about heterogeneity of disease, really at least in these two particular diseases, psoriasis and eczema or atopic dermatitis, folks have mostly focused on clinical heterogeneity and molecular heterogeneity. So the way it looks to the doctor evaluating the patient and what we look at like for things like RNA-seq. Histologic heterogeneity is something that's not really been particularly well incorporated into clinical practice. And as a practicing dermatopathologist, we know that when we look under the microscope, one patient with eczema or atopic dermatitis looks potentially very different from another patient with the same diagnosis. There are histologic hallmarks which help you arrive at that diagnosis. But really, what does that histologic heterogeneity mean? And really, our goal with this project was, given the increasing number of therapies, so this is an example for atopic dermatitis or eczema, and the cost of these therapies, and we know that patients don't always respond equally to the therapies, can we bring this all together to sort of make clinical decision making and therapy choice more sort of immunologically based. And just to introduce these diseases, in atopic dermatitis, there's a certain clinical presentation. I won't dwell on it. There's a certain histologic presentation. I also won't dwell on it. But what we know from a number of different studies that depending on your age, your ethnicity, whether you have intrinsic or extrinsic atopic dermatitis, and your gender, that actually, although Th2 activation is a hallmark of disease, depending on which sort of category or characteristics you have, you may have varying degrees of activation of other pathways. And so this is that idea of disease endotypes. And really, current histopathological analysis in clinical practice cannot distinguish between these different endotypes. We can say, yes, you have atopic dermatitis or eczema, maybe you have something else, but we can't really drill down with current methods on these different pathways. And psoriasis is really similar. There are certain clinical findings which are typical of disease. If a biopsy is done, there are certain histopathologic findings which are typical of disease. But what we know and what we've learned over a number of years is that there's really a huge spectrum in terms of what drives any individual patient's disease. And so, again, the question that I'm going to hopefully address somewhat today is, can we use new molecular techniques like RNA in situ hybridization to really drill down on these different disease endotypes and inform the clinician the best way to treat the patient that's in front of them? And so here's sort of a formal hypothesis slide. I think this does help sort of clarify the work that we've been doing. But really the idea is that molecular heterogeneity in inflammatory skin diseases, eczema and psoriasis in this case, has some relationship to clinical heterogeneity. It looks different in one patient than the other sometimes. Histologic heterogeneity, although there are unifying features of disease, it may look different depending on the patient you have. And can we somehow unite this with why patients don't always respond equally to the same therapy? And so we thought that maybe we could develop biomarkers that could help us with this. They might help us in challenging cases. I won't talk about this too much, but occasionally it's hard to tell whether a patient has eczema or psoriasis. There may be overlapping features clinically, there may be overlapping features histologically, and how do you choose the right patient or the right treatment for this patient, especially when they have severe disease? We want to look at molecular subclassification within each disease, this idea of endotypes, but ultimately in clinical practice, we want to help treatment selection. Our sort of bias going into this was that if we really wanted people to use this tomorrow or next month or next year, it really would be a technique that did not rely on sending a specimen to a laboratory, because there's a number of reasons, but ultimately dermatologists, when they're unsure, will do a biopsy. A dermatopathologist will get the tissue. And we really wanted something that would be in the hands of the dermatopathologist to interpret and help the clinicians out directly, as opposed to a send-out test or a test where someone may not have the expertise to do it or interpret it, or something that was very expensive. And again, eczema and psoriasis are common. You saw the number of FDA-approved therapies on the prior slide, and we really just wanted to evaluate it in these diseases, which seemed like a nice opportunity. And so if we take a step back, really, I think the first premise that we need to establish is that we can use disease biomarkers to actually distinguish between psoriasis and atopic dermatitis. And so what are the best markers? So we reanalyzed, which was at the time the largest study looking at cases of atopic dermatitis and cases of psoriasis by RNA sequencing. You can see the numbers here. It's a reasonably sized study. What really stuck out to us when we look at this volcano plot, looking at differential gene expression in eczema or atopic dermatitis or psoriasis, that not all, but many of the most differentially expressed genes were treatment targets. So in eczema, IL-13 and IL-4, IL-31 are all FDA-approved targets. In psoriasis, IL-12 B gene, IL-17A, not interferon gamma, but IL-23 are all FDA-approved therapies. Now, IL-36 family members are also approved for pustular psoriasis. So it really stuck out to us that a lot of the markers were actually the targets of therapy. and this was really intriguing to us. You can see this NOS2 gene here, which encodes iNOS, which we'll come back to, really stands out as a very strong psoriasis biomarker. And we weren't the ones to necessarily discover this, but certainly our findings were consistent with what was in the literature. So then the question is, how do we detect the markers? And as a pathologist, I think the default is immunohistochemistry. I also did a PhD. We did a lot of immunohistochemistry during my PhD. But it turns out, and I'll show you some of this data, it's really hard to detect cytokines and secreted molecules for the most part, especially when they're coming from immune cells by immunohistochemistry. Maybe the antibodies aren't as good, maybe it's another issue. And so we tried immunohistochemistry when we first started this. And again, I'll show you some of this data. But really, this is what led us to the possibility of using RNA in situ hybridization to detect these genes. So when we think about potential advantages and disadvantages of this approach, the thing that I've been just blown away by with this whole endeavor is how specific RNA in situ hybridization is for the markers that you're looking at. It's spatially resolved, which Denise mentioned is really kind of helpful from a research standpoint. It's really fascinating as someone that's interested in immunology and pathogenesis of disease. And just like the molecular instruments HCR assay, you could do this with a commercially available kit. And so we literally started by just ordering these kits online, and really were just blown away by the results that we got. As a dermatopathologist, all of our specimens, 99.9% of our specimens come in and are formalin fixed and paraffin embedded. And so the kit worked great with that. I'll also mention BioCare, who has really been a great partner in terms of their Encore Pro stainer and rolling this out as a laboratory developed test, which I'll talk about more. But I think the really important part of this in terms of adoption is this is something that dermatopathologists are accustomed to doing. They see a case, they have a question or a differential diagnosis, and they order a stain. So for all intents and purposes, it's very similar to ordering immunohistochemistry. And our intent in doing this was really to empower pathologists to help their dermatologists and ultimately help their patients by addressing these kind of questions. The potential disadvantage is that we're detecting RNA, not protein. All the markers that I'll show you today, the RNA correlates pretty well with the protein. That's certainly not the case for every last marker, but at least in the cytokines and other immune signals that we've been interested, looking at RNA has been very good. So again, coming back to that question, can we just, looking at markers, tell the difference between eczema and psoriasis, two diseases that have very distinct immunology, which are separate? And so we put together this cohort of patients with psoriasis and atopic dermatitis. We had patients with normal skin. And we really put together a panel of markers based on that RNA sequencing data that I showed you, looking at cytokine biomarkers of disease. And really, remember, in the upper left-hand corner of the slide was that NOS2 or iNOS protein. Our hypothesis was, based on the RNA-seq data, and we're detecting mRNA, that in psoriasis, this should really be through the roof, but not really in eczema or atopic dermatitis or normal skin. So I won't spend a ton of time on this slide. I'll show you higher power images on the next slide, but here are the H&E hematoxylin and eosin stain slides. This is how pathologists look at tissue for diagnosis. We can see a representative case of psoriasis, atopic dermatitis, normal skin. This low power image is a little bit hard to see the red chromogen. Again, I'll show you higher power images. But in this pseudo-colored plot where we're having each cell be a dot, you can see that in the psoriasis samples, we're seeing a lot of NOS2. In the eczema or atopic dermatitis samples, we're not seeing that. And in the normal skin samples, we're not seeing that. Here's a little higher power image of what this actually looks like under the microscope. You can see both the live H&E as well as our pseudo-colored interpretation. But what was really gratifying about this, and I must say a little bit surprising that this works so well, is every single case of psoriasis labeled for this marker that we just found, not randomly, but arbitrarily somewhat in terms of which case we were looking at. We had very sort of stringent methods in terms of how we had a validated case of psoriasis, so it was worth including. But none of the atopic dermatitis cases, none of the normal skin samples had any staining. And the medical student that was working on this at the time, medical students often have a difficult time with histology and these kinds of tests, understandably, because it just takes time to get good at. I mean, they would just immediately know. They'd see red or they wouldn't see red. And so I think that really speaks to sort of the clarity with which this test worked. So then we wanted to look at cytokines. NOS2 is not a treatment target in psoriasis. It's a nice biomarker, but it may not necessarily help us out with personalized medicine per se. And so looking at cytokines, they're actually approved treatment targets for psoriasis. So if a patient has bad psoriasis, a dermatologist may prescribe them a biologic medication, which is an antibody that actually blocks IL-17A. And so for us, this was very compelling. Can we detect this in the patient's skin? And again, sort of a similar setup here where we have H&E across the three conditions on the left. We have the actual chromogenic RNA in situ hybridization, which may be a little bit hard to see in the middle. And then we have those pseudo-colored plots, and hopefully you can appreciate there's red in psoriasis, but there's not red in any other condition that really confirmed our hypothesis. And really the staining is just so nice to look at. You can see that there are individual T cells, and we confirm this in a number of different ways. They are mostly T cells that are producing IL-17A, and they're in the epidermis, and that's where psoriasis pathophysiology is. So just very satisfying. It made a lot of sense as someone that's interested in immunology. In case of eczema, we didn't really see any staining, so you don't have to understand the H&E, but just know that there's no red staining. And when we looked across cases, we noticed that in general, in the epidermis and psoriasis, cases were positive. In atopic dermatitis, we did have a couple cases that showed a little bit of staining. In normal skin, we didn't have any. And so what I would say based on this data was that we're really excited that all psoriasis cases stained. We were intrigued by the fact that there's this variation in the amount of IL-17 producing cells in psoriasis. So remember this idea of personalized medicine. Maybe this is actually telling us something about these patients. And in atopic dermatitis, occasionally you may see some expression in the epidermis or the dermis. And again, we were just really, really excited about this. I mentioned during my PhD, I did so much immunohistochemistry. I stained hundreds and hundreds of slides, and it's probably one of my favorite techniques in all of medicine and science. But I've come around to really appreciate RNA in situ hybridization, because when we know psoriasis, so this is a case of psoriasis, is a T cell dependent disorder, and I'm showing you CD3, which labels T cells in immunohistochemistry. We can see the T cells in this disease. So there's some in the dermis down here, the paler part. There's some T cells up here in the epidermis, the bluer part. And our group tried out every commercially available IH-17 antibody we could find when we started doing this work. And when you see all the tissue turn brown, and we know that it's a T cell derived signal, and we see keratinocyte staining, we see fibroblast staining, we see endothelial cell staining, it's just really unsatisfying as a diagnostician and as an immunologist. In contrast, when you look at the RNA in situ hybridization, you see distinct individual cells that are producing the cytokine, and they're often in the epidermis. And actually, in most cases, they're in the epidermis. And this is just really intrinsically satisfying because it makes sense immunologically. And that's really where we got excited about this when we compared the staining to our IHC assays. So on the other side of the coin, atopic dermatitis or eczema is an IL-13 or Th2 type 2 dominant immune disease. And so we kind of asked the same question. Hopefully you can appreciate that whereas where we're looking at IL-13 in psoriasis, there's no staining. When we're looking at atopic dermatitis, there's plenty of staining. And when we're looking at normal skin, there's no staining. And although I didn't emphasize it before, the fact that we don't really see any staining markers in normal skin is just really, really useful and helpful. Here's a higher power image of what the staining can look like. Again, we're seeing individual cells that are staining red because they're producing IL-13. A diagnostic factor that dermatopathologists look for when they're diagnosing eczema or atopic dermatitis are these clear spaces. So hopefully you can appreciate the clear spaces in between the keratinocytes. We call that spongiosis. Where there's more IL-13 production, there's often a lot of spongiosis. So again, we're seeing T cells, and we prove this in other ways. We know from molecular studies that IL-13 is really important in atopic dermatitis. Our whole tissue isn't turning brown or red depending on the chromogen you're using. And we're actually seeing these cells increase in areas where we're seeing the pathogenic hallmark of disease, which really, again, just like this really seems like it's working. We didn't really see much staining in most cases of psoriasis. I won't belabor the quantification, but again, there's this idea that there's heterogeneity. Interestingly, not all cases of atopic dermatitis had IL-13 staining. Some cases of psoriasis had it. Thankfully, not many cases of normal skin had it. But doing these kind of projects has really sort of forced an evolution in the way that we think about psoriasis and atopic dermatitis, where some people think them as being diseases on one end of a long spectrum. And maybe a lot of cases are in the middle, and that heterogeneity sort of drives why patients may not all respond to the same therapy. So we looked at a bunch of different cytokines. These are mostly treatment targets, other cytokines of interest. And in terms of our laboratory developed tests, I should point out that this work is all published. I had the relevant citations before. In terms of our laboratory developed tests, which I'd be happy to talk a little bit more about how actually we enacted that in the question and answer period, we felt that in terms of the question of is it psoriasis or eczema, if it's eczema or psoriasis, what treatment is a patient likely to respond to, that this group of 4 cytokines was most useful. So there's probably not too many dermatologists in the audience, but in case there are, or dermatopathologists, will often order direct immunofluorescence. So that's a panel of markers that helps us evaluate for immunobolus disease. This is a very similar panel that helps us further characterize patients with eczema and psoriasis. And so there's 4 markers: interferon gamma, which tells us about type 1 or Th1 immunity, IL-13, which tells us about Th2 or type 2 immunity, and those couple psoriasis biomarkers. And we've come up with a way that we do the staining, and we're actually issuing reports with this information on it that are going back out to our clinicians. You can see some examples of how we do this on a cell-by-cell basis. This was more of a research sort of angled paper. We're coming out with a clinically oriented paper in terms of sort of how to do this for other institutions that may be interested in. But hopefully what you can appreciate is that it's pretty straightforward to interpret the signal that's coming out of these cells. And so how have our clinicians at Yale and now other places been using this? Well, there's a number of different settings where people are finding this useful. I'd say mostly it's this question of eczema or atopic dermatitis versus psoriasis. And what we know about treating patients with severe atopic dermatitis or eczema and psoriasis is that if we get the diagnosis wrong and we give them a medicine for, let's say they have psoriasis, but we give them eczema treatment, not only may they not improve, they may actually get worse. And these are medications that take a lot of time to get approved from our office staff. They're very expensive. And so this is a decision that now that we're in the molecular age and treating these patients with highly specific therapies, we really don't want to get it wrong. Dupilumab is a medicine that's commonly used to treat eczema or atopic dermatitis. We found some utility in this setting. I just want to make sure we stay on time, so I won't go into that too much further. Erythroderma is where the patient's skin is all or mostly red, and sometimes diagnosis and treatment are a little bit elusive. This thing called cutaneous T-cell lymphoma. Basically, other severe skin diseases that either are hard to diagnose or don't have FDA-approved therapies, our clinicians are asking us to use this test. And there's a number of other markers and a number of other settings which we're evaluating this in. And so I'll wrap up, let's see, in maybe 4 or 5 minutes. The first part was sort of about diagnosis. The other question is like, can we actually provide treatment relevant information? And so this is a really simplistic hypothesis. There's nothing really particularly sophisticated about this, but the idea is if the patient expresses the target of the medicine they're taking, they're more likely to respond to it. But if they don't express that target, they're less likely to respond to it. So we'll focus just for sort of an anecdotal example of this in eczema or atopic dermatitis. There now are a number of FDA-approved therapies for atopic dermatitis. So JAK inhibitors are oral molecules. They inhibit a large number of cytokines, including IL-4 and IL-13. Remember, IL-4 and IL-13 really are thought to be the keys of this, particularly IL-13. Dupilumab is an injected medication which inhibits both IL-4 and IL-13. Tralokinumab and lebrikizumab are newer medications which inhibit IL-13. They're all FDA approved, and you can see that IL-13 is sort of the hallmark that unites all these therapies. And so we thought that if you had eczema or atopic dermatitis and you didn't express IL-13, well, maybe you wouldn't respond to these therapies. Concomitantly, if you had a lot of IL-13, maybe we were right on track, we got the right target, and you would respond. And so we went back and put together a cohort of 61 patients with atopic dermatitis. We've come a lot further since this publication, but this is what's published. These are all patients treated with dupilumab, which blocks IL-4, but importantly IL-13. We had a skin biopsy, and we knew how they did on the medicines. That's how we found the patients. We classified them as either complete responders, you would never know they had eczema, partial responders, or non-responders. They may have gotten better, they may have actually gotten worse. And what we looked at is whether the staining actually correlates with the response pattern. So CR, complete responder, PR, partial responder, and a non-responder. And really, I was actually surprised that this worked. I was quite surprised that this worked. But what we saw was that when there's a lot of IL-13, patients are more likely to respond. When there's very little IL-13, they're less likely to respond, at least on sort of a population level. And when we saw alternate non-canonical immune signals like type 1 or Th1 immunity, interferon gamma, type 3 or IL-17, IL-23 axis immunity, that these tended to be patients that didn't do as well on the medicine. So really this kind of lends itself to this idea of disease endotypes. Not every patient with eczema or atopic dermatitis is the same. This differential immune activation, which we can't tell by histology or by looking at the patient, but which we can detect using this method, is actually meaningful. And so that's really where I'll stop today. We're very active in this area. We've got a number of other biomarkers which we're looking at. We're really looking at working on sort of validating this further, these approaches further in larger cohorts. But really, this is the first laboratory-developed test that I've been brought on in my career, and it's been really sort of neat and interesting to be a part of it and see how it's changing practice at Yale, where a lot of our clinicians are asking for it, but now more and more other institutions are either adopting this approach internally or sending us consultation cases to help them out. And so just in the last minute or two, I would just say that not only is the translational revolution coming to dermatology, it's coming to every medical therapy, and that our treatments are really highly targeted. And there often is this disconnect in terms of identifying, looking at a patient clinically, histologically, if you have that opportunity, and then this idea of disease endotypes or differences within each disease and how it affects therapy. And I think this problem is going to get more and more complex over time. And so our hope is that tests like this will help sort of navigate this changing practice. Hopefully I've instilled upon you somewhat that many inflammatory skin diseases show clinical and histologic heterogeneity, which we think is important in immunologic heterogeneity and treatment response. I guess I'll really close there. I would be happy to answer any questions during the Q&A period, but this has been it's been really fun to work with Anish and Megan and Joyce and others, Molecular Instruments. They make this collaborative sort of work really, really, really fun, as well as the folks at BioCare, including David Anderson and others. It's been a pleasure to work with them. They've made everything really easy as we've tried to sort of, roll out these laboratory developed tests. And with that, I'll stop talking, but I'd be happy to take any questions during the Q&A period. Thank you.
Thank you so much to both Dr. Acharya and Dr. Dansky for those insightful presentations. We appreciate you sharing your expertise with us today. At this time, I'm going to transition us into our Q&A session, and joining us now is Dr. Jason Ramos from BioCare, who will be answering questions alongside Dr. Acharya and Dr. Dansky. So I see that there are some questions in the chat. I'll pull up the first one. Dr. Damsky, this one's for you. How did you go about transitioning this from a research assay to an LDT?
Thanks. Yeah. So, we were blown away by how much this worked. And actually, my research program initially was in a very different area. But this just seemed to work so well that we felt like we couldn't ignore it. And so, as pathologists and dermatopathologists particularly, we use a lot of laboratory developed tests, mostly which are immunohistochemically based. But occasionally there are RNA in situ hybridization based approaches which are used. The vast majority of them are not FDA approved. So there's actually reasonably good framework in terms of how to develop a laboratory developed test. And basically, without going into too much detail, you basically just need to stain a reasonable number of positive controls and negative controls and show that the staining patterns are as expected and sort of document that. And so it actually was surprisingly, we did all the work and, checked all the boxes in terms of rolling it out, but actually it was surprisingly easy to actually start using this clinically.
Thank you so much. I think there is another question. This one is also for you, Dr. Damsky. The question reads, do you plan to seek for reimbursement?
Yes, so we do seek for reimbursement. So we use a code that allows us to bill for RNA in situ hybridization. So I should step back. And so we get a lot of biopsies where the clinician's asking for eczema or psoriasis. Or something else, like rule out this or that, other inflammatory disease. The dermatopathologists here at Yale do not reflexively order this on cases that are sort of within that spectrum, psoriasis, atopic dermatitis, because there's so many of them. So this is our current practice. We've all the clinicians at Yale are aware that we can do this, and increasingly clinicians at other institutions are aware that we can do this. And so usually we'll leave it to the clinician to ask for us to do this testing. And so they say, it's really important that I know if this patient has eczema or psoriasis because I need to know how to treat them, or there may be other related questions. We leave it up to them to ask. So it's all coming from the clinicians. This information would be really helpful to help me take the best care of my patient possible. And so in that setting, we are doing the testing and we are billing for it. And in the vast majority of cases, we get reimbursed through the patient's insurance. And I think it really adds a lot of efficiency to care. And especially when you think of the cost of doing this test versus the cost of the medication and the risk that the patient may not improve or get worse if the wrong medication is selected. And for those that may not be aware, some of these medicines are tens or even hundreds of $1,000s per year. And so just having a little bit more confidence that we're choosing the right medication going into the process can be really helpful. But to simply answer the question, yes, we are getting reimbursed in most cases for the testing.
Thank you so much. I think we have time for one more question. I'll pull it up right now. This question reads, are there other opportunities to improve patient care through development of similar LDTs for other diseases?
Yeah, thanks. I mean, I think this really highlights a really important premise of this test and sort of this whole area, and how easy BioCare and molecular instruments have made this for us, is that, I showed that picture of the iceberg in the beginning. I mean, if you look at all the clinical trials and the pipelines for dermatology drugs, there's just so many different therapies for so many different diseases. And this idea of disease endotypes and biomarkers of response is something that's, at least in tissue, relatively new to inflammatory disease, including inflammatory skin disease. If we look at cancer, which has been sort of a leader in sort of this area of biomarker development, I don't know what the current number is, but there's a large number of FDA-approved therapies looking at predictors of response to whatever targeted medication you may be interested in. I think, especially as these medicines are so expensive, there's going to be so many of them. There's going to be so many patients where dermatologists will be thinking about these medications, or outside of dermatology, that I think that having some grounded molecular evidence that a patient is likely to respond to whatever therapy is probably something that's going to be more and more of a requirement over time, even in inflammatory diseases. And so, again, I like that iceberg image, not to highlight my own work, but just to highlight the how complicated clinical medicine is becoming with as we know more about the pathogenesis of diseases and there's more and more approved therapies.
Yeah, just to add from our side to that, I mean, I think the coolest thing okay, there are a lot of cool things here, but one of the coolest things for me is that we didn't do anything special from our side. It turns out that the things that are important for this kind of workflow are actually pretty important for the research users too, which is low cost, high performance, good turnaround time. And so it's like just a testament to our standard process that we don't charge custom design fees. We do some of these things to try and lower the bar to entry. But even if we had some special workflow for these kinds of use cases, like we didn't use it here because we didn't even know. So I think that's one of the coolest parts, most validating parts for the way we do our job here.
Yeah, thanks, Anish. I just piggyback off on that, and I know we've got to go, but the turnaround time is great. So if a clinician asks for this on a Monday, sometimes we have information on Tuesday or Wednesday, and we can get this information to them in a clinically relevant turnaround time. And sometimes there's patients in the hospital that are sort of in an acute state of duress where we really need to know what they have, and we're able to turn these results around really quickly. And yeah, thank you for mentioning turnaround times. It's really important.
Thanks, Bill.
Yeah, so that wraps up our Q&A session. Thank you all so much for your thoughtful questions, and a huge thank you to Dr. Acharya, Dr. Dansky, and Dr. Ramos for sharing their time and expertise with us today. We hope that you found the discussion valuable and informative, and if you have any follow-up questions or would like more information, feel free to reach out and we'll be happy to connect you with the right resources. Please be sure to keep an eye out for Molecular Instruments social channels in your inboxes, and the on-demand link will be sent over shortly. On behalf of our entire team, thank you again for joining us, and we look forward to seeing you at a future webinar. Have a great rest of your day.
Thank you, everyone. Bye.
Thank you.